Good afternoon, everyone. We are the Plastics Spring 2016 research team, and for this past semester, we've been quantifying plastic in our oceans. I'm Sophie Meyer.
I'm Will Jackowitz.
I'm Zoe Maud Alec. I'm Lizzie Feldman DeMello.
I'm Cooper Allen. Charles Whiting.
Ron Thomas.
Thank you. Now, for our introduction, I'm going to give you a little bit of background. Now, a study predicts that plastic will outweigh fish in our world's oceans by 2050. Yes, at the predicted rate of production, if failing to properly dispose of plastic, it is expected to outweigh fish in our world's oceans in less than 35 years. In fact, the worldwide use of plastic has increased 20-fold over the past 50 years, and is expected to double that in just the next 20. And with an increasing world population, there is only a greater demand for this material we call plastic. But about 1/3 of all waste ends up mismanaged, and what this looks like is about 4.8 to 12.7 million metric tons of plastic entering our oceans annually. And what this plastic does, constantly exposed to the sun, is that it breaks into tiny pieces through a process called photodegradation. And these small microplastics have big effects.
Some of the effects of plastic pollution are well documented. Here we have a seal, a trout, and a turtle all entangled in plastic pollution. And on the bottom right, we have an albatross bird rotting away on the beach with loads of plastic in its stomach. However, what we're about to discuss today is not well documented. We are looking at plastic ingestion by economically important fish, which are drivers of the Bahamian economy. The effects of plastic ingestion not only hurt the fish, but they are also incredibly detrimental to human health. Plastics act as vectors for POPs, or persistent organic pollutants, which can be DDT, PCB, flame retardant, and a number of other harmful chemicals. They are hydrophobic, meaning they do not mix with water, so they bond to the plastic, and when it is ingested by fish, it is then lipophilic, so it gets absorbed by the fatty tissues in the fish's stomach. And when we are eating fish, we could potentially be ingesting traces of these chemicals. They can be carcinogens, they disrupt hormones, they increase diseases, and they can cause birth defects. In studies with birds, plastic POPs significantly decrease the eggshell thickness, which led to sharp population decline in birds. And in humans, they can be mutagenic and tetragenic, meaning they cause mutations on the DNA level.
So our research questions are fish of economic importance consuming plastics? What is the density of plastics on the surface of the Exuma Sound? And is there a connection between the plastics ingested by fish and the plastics found in the epipelagic layer of the water column? That layer of the water column being the topmost layer. So our research objectives are to analyze the quantity of plastics ingested by different fish species, analyze the density of plastics on the epipelagic layer of the water column, and to then categorize these plastics found by size, color, and type.
So I'm going to talk a little bit about our methods. So we were focusing on finding plastic in the pelagic game fish that were feeding on the epipelagic layer of the water column in the Exuma Sound. Now the Exuma Sound and the southern tip of Eleuthera are an inlet for the Atlantic Ocean, and currents are coming into this inlet at great strengths, and they're bringing in plastic with it. This plastic is easily drawn into this inlet, however it's prohibited from leaving this inlet. So we would go out about once or twice a week and catch fish such as mahi-mahi, tuna, wahoo, and the like. So we would also conduct trawls while we're out on the boat. So as you can see here, this picture is the trawl, which is a big net that gets dragged behind the boat, and it picks up plastic on the surface layer of the water. So back in the lab, we would conduct dissections. So basically we would cut open the stomach and we would empty the contents into a stack of sieves that ranged from 4,000 micrometers to 63 micrometers. So water was then poured over these contents to make sure that they were in their respective sizes. Then the layers would be taken off and we would check to see if there was plastic present. So if there was plastic present, we would determine the quantity of the plastic found, the type, the color, and the size of the plastic. For the trawls, this was also applied. So we would check to see how many pieces of plastic were in the trawls and what color, type, and size the plastic were. To determine the density of the plastic in the trawls, we would take the speed at which the trawl was going and the distance traveled.
So now the results from our studies. Our data set included 203 fish from 11 different species, 4 of which had ingested plastic. These being the Atlantic flying fish, the kingfish, the mahi-mahi, the wahoo. Of the fish that ingested plastic, the mean amount was roughly 3 pieces. And with all this information, we deduced that the percentage of fish that had ingested plastic amounted to 15.8%. In every single trawl we conducted, we found plastic. The mean amount being 15 pieces, and the range being 2 to 35 pieces. With this information, we concluded that the mean density of plastic on the surface surrounding Cape Eleuthera was roughly 74,500 pieces per kilometer squared. As you'll see on this color scale graph here, we have red the most dense locations of trawls as far as plastic on the surface, and blue being the least dense. One thing to take into account is the observation that the plastic on the surface is typically more dense around this convergence zone known as the wall. So the categorization of the plastic we found. On the y-axis, you'll see the frequency or the abundance of plastic, and on the x, you'll see the plastic type, plastic color, and the plastic size. For plastic type, the most frequent found in trawls were the fragments, whereas the most frequent type in stomachs was the film. For both trawls and stomachs, the most frequent colors were clear and white. In both trawls and stomachs, the most frequent size was 0 to 0.39 centimeters. We conducted a ANOVA test for each of these graphs. Each yielded a p-value less than 0.01, meaning that there was in fact significant difference between each of these categories. Finally, a t-test was conducted between trawls and stomachs. However, the p-value was greater than 0.05, meaning there was not in fact statistical difference between the two.
And now on to the discussion section. This study clearly determined that plastic can be found in high densities within economically important game fish, as well as in the epipelagic layer of the water column. This likely being a result of bioaccumulation, and that's the transfer of plastics up the food chain through predator-prey relations. And though that doesn't have an immediate effect on the health of humans, the POPs that are attached to these plastic pieces that assimilate themselves in the tissue muscle of these said game fish do have effects on humans. Yet the only evidence we have for this claim, is the dissection of 2 prey fish in this study, 2 Atlantic flying fish to be specific. And of these 2 prey fish, 1 had plastic in its stomach, equating to 50% of the prey fish in this study having plastic in their stomachs. And though this sample size is very insignificant, of that being only of 2, it is likely an indicator that prey fish retain plastic for the beginning process of bioaccumulation. Interestingly enough, in similar studies conducted in 2013 and even further back in 1970, documented similar plastic ingestion rates of 19% and 14%, ours being 15.8%. Through analysis of trawl data, it was clearly identified that plastic can be found in higher densities closer to the wall off the southern coast of Eleuthera rather than farther away from the wall off the coast of southern Eleuthera. I'm sure you're all familiar with by now, but the star indicates where we located are currently in the boathouse. This wall is shown through the different shades of blue. What we found, or hypothesized, is ocean dynamic forces of plastic into convergence zones, this being above the continental shelf. Ocean dynamic forces consisting of tides, currents, winds on and offshore, and upwelling forces. Upwelling forces being nutrient-rich, deep ocean currents that travel along the bed of the seafloor until continental shelf, in this case, or a landmass, forces up to the surface. White translucent pieces of plastic ranging from 0 to 0.39 centimeters in length were the most abundant in trawls and in the dissections of fish. And though this is interesting to note that white translucent pieces of plastic were the most abundant in fish and on the epipelagic layer of the water column, it is unlikely that these fish are targeting these specific pieces of plastic due to their color, due to their passive feeding techniques that we find in prey fish, such as Atlantic flying fish. Rather, due to UV exposure and other weather conditions that break down these plastics, as already previously stated, these plastics lose their pigment and then become white and translucent, having more white translucent pieces of plastic on the epipelagic layer of the water column and then in the stomachs of these fish.
So these plastics, as previously stated in this presentation, act as vectors for POPs. These POPs being harmful chemicals which in turn deplete the integrity of the biodiversity in marine environments. Moving forward, we need to establish legislation which either cuts off the production of single-use plastic or monitors the way we deal with our waste, because as stated in our introduction, up to 1/3 of our waste is mismanaged and ends up in our oceans. There also needs to be more awareness regarding this issue, so we need to educate those who aren't aware of how prevalent this is as a topic. Some future studies to further our current study include quantifying plastic in Atlantic flying fish, like prey fish in general, setting up satellite buoys in the Exuma Sound so we can determine the current patterns to get a more accurate estimation of where plastic travels and its tendencies. And we would also hold a captive study with mesopelagic gamefish to determine their retention rates to see if the plastic passes through their digestive systems or if it assimilates into their body tissue. Thank you. We would like to acknowledge the Cape Luther Institute, Zach Zuckerman, Holly Bursch, Rob Drummond, Annabelle Brooks. Photo courtesy to Zach Zuckerman, Dr. Owen O'Shea, the Five Gyres Institute, and Miguel Furtado. Here are our references. Thank you, and are there any questions?
Is there any type of digestion that happens when the fish ingests the plastic, or is it all stuff that doesn't get passed through and eventually gets into the muscle tissue?
Thank you. That's a great question. So the question was, is there any passing of these said plastic pieces in economically important game fish, or do they all assimilate into the muscles of these game fish? So as we already discussed, this would hopefully be a future study that would tackle this research question. Yet it isn't really the most important, seeing that these plastic pieces act as vectors for harmful chemicals, POPs, and those do assimilate themselves into the muscle. So though plastic can cause blockages and bruising internally, what really is detrimental to the fish and then eventually humans is the POPs that are assimilating themselves into the tissue that we already have determined.
I have a question. Great, so you say it was on average about 74,500 pieces per square kilometre of plastic in the Exuma Sound. This is incredibly high, and it's a lot higher than a lot of other previously published estimates. Could you explain why this figure might be so high, and how accurate are you in that extrapolation?
Right, so the question is regarding our information that stated that the mean density of plastic on the surface surrounding Cape Eleuthera was roughly 74,500 pieces per kilometer squared. How accurate is this data, and why is this so high in comparison to other studies? And to answer that question, we hypothesized that Northern Atlantic subtropical gyre forces plastic in this region in particularly high densities. And for example, this particular region, a lot of convergence zones, such as landforms such as this island, Eleuthera, as well as the wall, as discussed earlier, will use upwelling forces to create these higher densities discussed. So in our particular situation, the density is particularly high because we conducted roughly half of our trawls near this wall, which is a convergence zone for plastic.
Have you guys seen any physical evidence of damage to the stomachs or internal organs of the fish?
Great question.
Thank you. That was a good question. And that was, have we seen any physical damage to the insides of the stomachs? Now, when we dissect the stomachs, we have not found, in our study at least, any obvious physical damage, because most of the plastic features we found were smaller, and we didn't see any puncturing either. But we did also cut through the stomach, so it would be hard to find if there was a tear, but we would also know if there was a tear previously established because of the contents leaked.